talk about the actual structure of DNA, I'm sure you've heard the acronym many times before, but do you actually know what it stands for? Well, it might seem like a bit of a mouthful at first, but I promise by the end of this screencast you'll have a better idea of what it means. But DNA stands for deoxyribonucleic acid. So let's look at deoxyribonucleic acid in a little bit more detail. Over here you'll see a picture that you may or may not be familiar with. It's a diagram that you might have seen before, and the most obvious feature and one of the most important features of DNA is that it seems to be these two strands that are twisted around each other. And in biology that has a name, and we call that a double helix. So DNA is a double helix, meaning that it's two strands twisted around each other. But what makes up each of these strands? Well, just like the carbohydrates, proteins, and fats that you learned about in the last unit, DNA is also a polymer, meaning that it's made up of many, many subunits. So each of those strands in the double helix is made up of many repeating subunits. If you recall, carbohydrates such as starch were made up of subunits of glucose, and proteins were made up of subunits of amino acids. DNA strands are made up of subunits of nucleotides. And this is what one nucleotide looks like. It's got three parts. The first is a phosphate group. The second is a sugar called deoxyribose. And the fourth is a nitrogenous base. Okay, so a strand of DNA is made up of many units called nucleotides. Each unit has a phosphate group, a sugar, and a base. And there are two strands that twist around each other that make a double helix. Let's look at it in more detail. First thing that you should be aware of is that there's actually 4 different types of those nitrogenous bases. So if we have our nucleotide here again, our phosphate, our sugar deoxyribose, and our base, there's actually, you can see 4 different kinds of possibilities. Our base could be cytosine, it could be guanine, it could be adenine, or it could be thymine. So those are the 4 possibilities you could have as nitrogenous bases. So the next question is, how do these nucleotides join together? You can see in this diagram that there's actually 4 nucleotides in this molecule of DNA. How are they joined? How are they linked? Well, through bonding. And that bond happens between the sugar of one nucleotide and the phosphate of the next one. So we call it a phosphodiester bond, and we say that DNA has a sugar-phosphate backbone because the outside of each strand of DNA is made up of those repeating units of sugar and phosphate linked by those phosphodiester bonds. But as we mentioned on the last slide, DNA is a double helix. So what about the other strand that's missing over here? How is it connected? Well, that strand is also connected by bonds, but this time it's hydrogen bonds, right? So the nucleotide or the nitrogenous base of one nucleotide bonds with the nitrogenous base of the other nucleotide through a hydrogen bond. Okay, so the sugar phosphate backbone is on the outside, linked by those phosphodiester bonds, and the nitrogenous bases link on the inside through hydrogen bonds, and that's how we get those 2 strands joined together. The really interesting thing about DNA is that those nitrogenous bases will only pair in a certain way. So for example, adenine will only bind with thymine, and cytosine will only bind with guanine. And we call this complementary base pairing, and it's really important, as you'll see later on. Another thing you might notice is that this strand is the opposite of this strand, right? So it kind of looks like it's upside down. That's another important feature of DNA, and we call those strands anti-parallel. So let's do a little bit of practice with the structure of DNA. Alright, well let's start off by labeling what we already know. So we can see here we've got one nucleotide. Okay, so one nucleotide. And our entire DNA strand is, in this case, about 5 nucleotides. One nucleotide is made up of a sugar called deoxyribose. So we've got that. It's made up of a phosphate, and a nitrogenous base, which in this case looks to be adenine. And we know that each of the nucleotides are linked by these phosphodiester bonds. Okay, how do we make the anti-parallel strand to this DNA? Let's have a look. Well, we've got adenine here, and because of complementary base pairing, adenine will only pair with thymine. We've got cytosine here, which only pairs with guanine. We've got guanine here, which only pairs with cytosine. We've got thymine here, which only pairs with adenine. And we've got cytosine again, which only pairs with guanine. Okay, so that's how you would create a double helix, and hydrogen bonds would be joining up the nitrogenous bases. Those are the main features of DNA. If you'd like more practice, there are some links provided here. Now, do you think you could draw and label a simple diagram? I'll help you out to start. The first thing you're going to want to start with is, let's start with our sugar molecules. So let's draw our little pentose sugars. Let's use 4 nucleotides for this diagram. Okay, so there's my deoxyribose. The second thing I'm going to add are my phosphate molecules. So there's 1. And the last thing I need is a base. Okay, and now I know that I need to join my nucleotides on the outside using those phosphodiester bonds. So let's join them up. Okay, now I have to attempt to draw the anti-parallel strand on the other side. So I'm going to start again with my sugars. And I'm going to add my phosphates. And my nitrogenous bases. Okay, and I know there are some hydrogen bonds that form between here. And I need my phosphodiester bonds as well. And there you have it, a very poorly drawn diagram of DNA. Can you label it? Let's see. What are the important features? Well, we can start off by saying that we've got a double helix here. Whoops. And that our double helix, these 2 strands are anti-parallel. We can also label that each of these structures is 1 nucleotide, and that the nucleotides are joined by phosphodiester bonds. And that the nitrogenous bases are joined by hydrogen bonds. We can also label that this is, let's say this one's adenine, this one is cytosine, which would make this thymine, and this would have to be guanine. We can label this, let's label this sugar as deoxyribose. And the last thing I can label is my phosphate. And there you have a drawing that's labeled of a simple diagram of DNA. Before we talk about how DNA replicates, let's review why DNA needs to replicate. Well, the reason is because before cell division, we need to double the quantity of DNA we have in our cells to ensure that each daughter cell has the appropriate amount of DNA, or a copy of each DNA molecule. And as you'll remember, this happens during interphase of the cell cycle. So what are the steps in DNA replication? Well, in part 1, we first need to unwind the double helix into 2 single strands, and that's done through the help of an enzyme called helicase. Helicase initiates the separation by starting at one end of the DNA molecule. You can see there's the enzyme helicase here. And it moves one base pair at a time, breaking those hydrogen bonds between the nucleotides. And it does so that the double-stranded DNA becomes single-stranded, 2 separate strands. Let's see this process in action. So here we have our DNA helicase molecule and our double-stranded DNA. The DNA helicase moves along the nucleotides, breaking the hydrogen bonds between the nitrogenous bases. And what we started with was 2 strands of DNA, which quickly become 2 single strands of DNA. In the next step, those now open single strands act as templates for the new strands. So the nitrogenous bases on each of the single strands are now unpaired. In the environment of the nucleus, or the nucleoplasm, there are many free-floating nucleotides. So we have adenine, thymine, guanine, and cytosine just floating around in the nucleoplasm that are available to form complementary pairs with those now unpaired nucleotides of the unzipped molecule. And so they come in and they form hydrogen bonds between the unpaired nucleotides, and that process is catalyzed by an enzyme called DNA polymerase, which you can see over here. So this enzyme here will catalyze the formation of the bonds between the nucleotides along here. So let's see that animated. Okay, so at this point our double-stranded DNA has now been unzipped by DNA helicase to become 2 single strands. And as you can see, all these nucleotides here are now unpaired. They have no complementary base pairs. But, as I mentioned in the nucleoplasm, there are many free-floating nucleotides. So there's thymine, adenine, guanine, and cytosine just floating around, ready to form pairs. So these will match up. Thymine, of course, will bond with adenine, and adenine will bond with thymine. And the reaction between these new nucleotides will be catalyzed by the enzyme DNA polymerase. So the DNA polymerase will help to form those phosphodiester bonds between the nucleotides. Okay, so that is a phosphodiester bond here. Okay, and then the nitrogenous bases will also form hydrogen bonds. So there'll be 2 there. Okay, and then DNA polymerase keeps moving along the molecule, catalyzing those reactions again and again and again until we form a new strand of DNA that is complementary to the old strand. And it keeps going. Okay. This complementary base pairing is very significant in DNA replication. As you know, adenine pairs with thymine and cytosine will only pair with guanine. That ensures that the new DNA molecule that's being made is identical to the original. And as long as no mistakes are made, the sequence of base pairs will be conserved. So that sequence of nucleotides stays the same. So after all the new nucleotides have been added, the phosphodiester bonds have been catalyzed, the double helix reforms. So the daughter DNA molecules each rewind back into this familiar structure here. We call that semi-conservative DNA replication. The reason being is that the parent strand acts as the template for the new strand. So each molecule of DNA is actually formed by one old strand and one new strand. So we call it semi-conservative because that old strand is conserved from the original parent strand. This diagram here is showing you some other possible models, conservative replication and dispersive replication. If you want to know more about how DNA replication was discovered to be semi-conservative, you can read about the Meselson and Stahl experiment. They were the one that discovered this form of DNA replication. And you can also read more about Arthur Kronberg, who was the one who isolated DNA polymerase. So there's an animation here and some tutorials that you can try. And that's the end.